All right. Yeah, we are 15 minutes in now, so let's go ahead and get started. Very excited to have all of you here because, as you guys know, neuroscience is my main thing. I'm very excited about it. I always love an opportunity to talk about the brain. We're going to go through a lot of content today because the brain is the most complex known structure in the universe, and there is a lot about it that I want to tell you guys. But so just keep in mind that what we should understand from this is just the core concepts, because there's going to be a lot of terminology that I'm going to have to fly through in an hour and a half. And so just sort of understand the general principles if you can, and then we can worry about the terminology at another point. I plan on next semester taking this talk and stretching it out into three or four talks so that we can cover some more material. But all right, so today, I'm going to be covering a little bit of cognitive science in the beginning. Cognitive science is sort of the study of what it's like to have a brain. So in terms of what your experiences are, what we think consciousness is, and what that's like for us. But then neuroscience is more of the how does the brain actually work and what does the brain do. So we'll be going into some neuroanatomy, neurophysiology, and then at the end, a little bit of neuroprosthetics. But I put in a ton of content for these, more than I thought there would be. So the neuroprosthetics for this time will be just a little bit tacked on the end, but we'll focus on that more in the future. And everything that I'm going to be talking about, for the most part, all of the figures comes from this neuroscience book, which if you're interested in it, you can always borrow it. That's a great introduction to neuro. Actually, before we get into that, I just kind of want to set some context for the world in which the brain lives, right? Because the brain is, I like to think of the brain as a cheater. And what I mean by that is for us to exist in this world as humans in our body, we have to always be taking, in terms of like being in nature, our brains have evolved to help us escape certain scenarios and cheat the system a little bit. Because we're in bodies that are not very well equipped to be living outside and fighting bears and living in the elements. And so we learn how to adapt. We build tools, we create clothes to act as fur, we build a roof over our head, and we learn to use various things. So again, tools like a hammer, stuff like that. And our brain learns to incorporate all of these into its mind-body image of what it has. And this is a really, really important concept, because everything in its functioning is, in essence, cheating some sort of underlying phenomenon. And so what I mean by that is the brain is, I would argue, the most powerful supercomputer on the planet. But yet, when you compare it to like a calculator or a computer, we can say, well, this computer can do computations much faster than our brains can do. But that's not actually true. It just sort of is in a different domain and is trying to solve a different goal. But if we think of consciousness in general and what that is trying to do, it's sort of cheating the understanding of all of this data that's going around us. Because our eyes are taking in data, photon data of different wavelengths, right? And it's coming in just sort of like pixels on a screen. And same thing, our ears are taking in compression waves. And our touch, we're taking in pressure and temperature and all of these things. And these come in as raw signals. Just like a computer, you have on and off signals, and then in the brain, they're graded as well. But that's not what we experience subjectively. What we experience is consciousness, which is a little bit muddy and not quite a term that we even really know how to define very well right now. But what that's doing for us is allowing us to understand and interpret this massive amount of data that the brain is processing in a way that's sort of cheating what's actually going on underneath. And so if we think of something like emotion, which again kind of seems like a muddy concept, and for people programming computers and stuff, there's even the question of why would you want to have emotion programmed in. But in emotion, there is a tremendous amount of data that you're understanding about the situation that you are in, the state that your body is in. Maybe you're looking at someone else and your emotion is influenced by that. And your brain is taking in millions, if not billions, of data points to be generating these sort of experiences that we have. And all of these experiences come together within the brain to form, all at the same time, to form the emergent property of consciousness and have all of this information going on at the same time. And so in a sense, to kind of wrap up what I'm saying, consciousness and our just subjective experience of existing is a good way for the brain to take in this overwhelming amount of information and just be able to operate and move around without getting bogged down in the details of that. Actually, one last example that I like to talk about with that is in terms of vision. Like the brain is simulating everything that we're experiencing. And we only have, like if you put your fist out in front of you, there's about a 5-degree field of view that's really, really high acuity vision. Everything else, the farther you go out, the blurrier it gets and the less color vision that we have. But the brain, based on what it knows about the world around us and the information that it's picking up from microsaccades moving the eyes around, it creates this simulation around us that it feels like we have great vision all around us, right? And this is true with other senses as well. And so it creates the simulation, and that simulation can be easily influenced as well. If you have someone with a mental illness like schizophrenia, you have these top-down processes that are making the person feel and have an experience of something that is not actually there. And same thing you see with like damage to the brain. And we see how fragile this simulation that we're experiencing actually is. And so these are all things that, that I think are important concepts for us to understand about how the brain is working. This is at a very high level. This is more of the cognitive science piece. We're going to dive in now to the nitty-gritty of neuroanatomy, but I think it's important to keep all of that in context. Cool. So a lot of this stuff in the beginning might be review for some of you, especially those who have taken bio or have a biomedical engineering background. But our nervous system, which comprises of our neurons in our brain and all of the things shooting electricity around our body to make us function, is split into the PNS and the CNS, the peripheral nervous system, and the central nervous system. And so the peripheral nervous system is everything that's outside of the bone. Look at that, laser pointer works. Okay, that's cool. Everything outside of the bone. And so this is moving down your arms, allowing you to control your muscles. It's also collecting sensory information from pain and temperature and pressure, going back towards the spinal cord and towards the brain, and communicating with your organs. It's telling your heart to beat. It's making sure that your stomach feels okay and helping that process along. And so that's the important functions of the PNS. And then the central nervous system includes your spinal cord and the brain. And the easy way to remember that is that it is encased in bone. And interestingly, this is not just an arbitrary line that we draw, but if you go in and zoom in on a very, very small scale, the actual anatomy is quite different, and the physiology of how this stuff functions is quite different on a small scale. So just to rush through the PNS pretty quickly, there are parasympathetic and sympathetic nerves. There are a couple other pieces, but we'll just focus on this right now. So this is sort of your fight or flight response. So I'm focusing on this because this is like some of the unique processing that goes along with the peripheral nervous system. So sympathetic, counter to its name, is your fight response. So that's going to be doing things like stopping digestion, dilating your eyes, and making your breathing get a little bit faster. And the parasympathetic is the opposite of that, slowing down your heart rate and sort of making you calm again. So now we're going to move on to the central nervous system, which is where we're going to stay for the rest of this because the brain is super interesting. But we'll start with the spinal cord. So its main function is to transport motor and sensory signals from the body to the brain and from the brain to the body. But it does actually do some processing on its own, which is really interesting. So it does reflexes when you're at the doctor and the doctor hits your knee and your knee goes forward, or if you touch a hot stove and you pull your arm back. That information isn't actually making it to the brain until much later when you actually feel the pain. That is something that's processing that takes place in your spinal cord. Another cool thing is that the spinal cord coordinates motor activity. The brain is not actually controlling specific muscles. And so if you stimulate using electrodes in certain parts of the spinal cord, you can actually cause an organism to walk even without input from the brain. All right, so that's the spinal cord. So now we're going to focus on the brain specifically. There are three main parts that I'm going to focus on, which are the cerebrum or forebrain, which is this large, in humans anyway, this really, really large top area. The cerebellum, which is Latin for little brain and sits on the back. And then the brainstem, which is functionally distinct from the spinal cord, but is continuous into the spinal cord. And so this little line there is designating that we've switched into the spinal cord. Just so that everybody knows, this is a medial slice. It's a half slice of the brain. This is a full brain here. So this brain would be facing this way. The front of the person's head is closer to me, and that's a slice down the middle. Does that make sense to everybody? Is everybody good with that? Okay, cool. All right, so the brainstem is the oldest structure, evolutionarily speaking, that we have in the brain, but it does have a lot of very critical life functions. So this is heavily involved in attention and heart rate and body temperature, digestion, breathing, urination, sleep, and of course it connects the more superficial areas, the areas up on top, to the rest of the body as well. And so this is actually the most critical area. If you get if this receives damage, it's very unlikely that someone will continue to survive, at least with good quality of life. So, but thankfully, this area is well protected. The skull comes down to about here, and then here's like the face. The eyes are right there, so it is really well tucked away. The cerebellum sits at the back of the brain, just under the forebrain, and it actually has a ton of neurons of its own. The forebrain has about 86 billion neurons. The cerebellum has anywhere from 40 to 65 or so billion of its own. I put that range because we don't actually know right now, and this is a really important thing that I should mention, which is that everything that I'm talking about today is the current dogma, what people understand to be true right now. But the neuroscientist in me and any other neuroscientist could always do the, actually that thing might be this, or it might be this thing. But for right now, at least in basics, we're going to say whatever I say is true, nothing but the truth, and 100% true. So anyway, cerebellum is used for motor coordination, precision, and timing, and also possibly used in language and emotion as well. And so what does that mean, the motor coordination? So the upper parts of our brain tell our arm where to go, but this is sort of your fine motor skills. So this would be the difference between if I want to move my hand to my face and touch my nose, the difference between touching my nose and slapping myself in the face. That is thanks to the cerebellum. There are some animals like the elephant that have a huge cerebellum, which is due to the dexterity of their trunk. That is almost entirely controlled by the cerebellum. All right, so we'll be spending the rest of the time here in the forebrain, which comprises of the cerebrum, which comprises of the cerebrum up here, and then this central part of the brain, which we'll get into in a little bit. So again, this is a medial view, a slice. Front of the brain is over here, and this is showing a slice down the middle. We have two hemispheres here, the left and the right. Again, this is the front of the brain, the back, left side, right side. All right, so first question, what are the three main parts of the brain? Does anybody know? Yes?
Cerebrum, brainstem, and cerebellum.
Awesome. Do you want candy?
Sure.
All right, cool. Awesome. Okay. Yes, very good. Okay, so now we're going to go into the forebrain, like I said, and I'm actually going to switch over to a program so I can show you in a 3D model. I just have these slides just in case things don't work, but we are, all right, so everybody can see this, right? This is from a website called brainfacts.org. If you want to work on neuroanatomy on your own, you can go here. It's pretty easy. You just select the area that you want to go to for, say, the frontal lobe, and it gives you some information about that, which is really nice. But I said that we would start with the limbic system. So let's go down here. So this is in the center of the brain. We see that it is lateralized, which means that there is one on each side. It's an important thing to note with the forebrain is that pretty much everything is duplicated. They have slightly different functions, but there's usually two of everything, and we call that being lateralized. So I'm going to isolate this structure a little bit here so that we can see what it actually looks like. And the limbic system is really important in learning and memory, homeostatic functions, and emotional regulation, anger, things like that. So there are three parts. We've got the hypothalamus, which is very useful in, again, the homeostatic functions. The hippocampus out here, which is important for learning and memory. Actually, there's a really famous clinical case of HM. They've referred to the patient as HM. I'll get into that a little bit later, but his hippocampus was removed, and he was not able to make any new memories from that point on. He was in his 50s, I believe, when this lesion took place. It was because he was having epilepsy, but he was able to remember previous memories. So this is implicated in making new memories. And then the amygdala is referred to as the 4 F's in neuroscience: feed, fight, flight, and reproduce. So those are the 4 functions of the amygdala. What did I want to do next? All right, thalamus. Let's see. Also, if people have questions as I go through this, please jump in. So the thalamus is really important. It's not the sexiest brain structure, but it's these two walnut structure at the center of the brain, and it is essentially a relay system. Pretty much everywhere in the cortex and lower areas and sensory areas runs through this, and there are a ton of what we call nuclei, which you can see here, which are functionally distinct elements that are a part of this. And so there is a lot of information moving through this, and it is one of the more important places to target clinically, especially with neurotechnologies, because pretty much everything passes through there. All right, let's see if we have the basal ganglia. So the basal ganglia also at the center, I'll bring it back a little bit. Also tucked in at the center of the brain is really important for motor initiation, so deciding, okay, I want to move, and actually kicking that circuit off, as well as your reward structures are in here. So when you hear people talk about dopamine and the reward system, that's originating in the basal ganglia. This is a really common area to target with a technology called deep brain stimulation. So that's where you stick in an electrode to help treat disorders where someone might be addicted to something. You can help overcome that, or something like Parkinson's disease. Because of that motor initiation, you stimulate this area and then they have more volitional control. All right, so moving on to the neocortex. These are the very top layers of the brain, and we're only referring to about 4, 5, 6 millimeters of space up on the top. And this is where almost all of your higher processing, higher cognitive functions come from. And this, a large neocortex, is what separates humans from the rest of the animal kingdom here. And so importantly, before we get into that, we see that the brain is wrinkled. It's a common insult to call someone smooth-brained to mean that they're stupid, but what does that actually mean? Does anyone have any guesses as to why the brain is wrinkled? Any guesses? Yeah?
To increase surface area?
Yes, absolutely, absolutely, very good. Do you want candy? Pass the candy around. I'll put this one out in the thing too. Very good. Yeah, absolutely. So the role of this is to increase the surface area, as you said, because as I just said before, this is the most important area for higher level cognitive functions, the things that we would think of as being intelligent. And I don't know how well you can see this, but this is a drawing that I acquired from, I think it was somewhere around 1870 from Philadelphia General Hospital. This is a drawing of a slice of brain. And you can see that the very outside here is a little bit darker than the rest of that, right? And so this outside is the cortex, and the rest of it is white matter, which we'll talk about later. And so again, this kind of shows how important it is to have this folded structure. And so this shows to scale the size of the actual brain and then the size of the unfolded cortex. So you see that we really are getting a lot more neurons packed in there as a result of folding that. Very good. OK, so the cortex is also split into 4 lobes. So we will go back over here to our lobes and start with the frontal lobe. And so the frontal lobe takes up a lot of space. It's really, really big in humans. If you look at other animals, this is usually the biggest difference that you see between us and other animals, even primates. And that makes a lot of sense when you look at the difference in our intelligence, because this is highly correlated with emotional social processing, as well as executive functioning like mathematics and logical reasoning, things like that. There's also an area of the brain right here that we'll talk about later that's very, very important in language and is much more developed in humans than it is in any other animal. So this is a really important area for higher level cognitive functioning, what we call executive functioning. Let's see, yeah, and then the prefrontal cortex specifically is the part that really helps us make decisions, again, mathematics, social behaviors. This is very, very important for that higher level thinking, and that's a subset of the frontal cortex. There are a bunch of other areas in here as well. We'll talk about some of them later. We'll talk about the motor cortex, which is at the very back of the frontal lobe, which is involved in making movements, but we'll get there in a little bit. Moving caudally, so we'll talk about that later. Moving back to the back of the brain, we have the parietal lobe. Parietal lobe is involved in sensation. So there's an area here that this is where all of your feeling, touching, hot, cold, pain, all of that comes from. And then in the back, this is really important for sensory integration. So this is a starting point for helping us understand, again, in our conscious experience, have vision and hearing and touch, and all of that makes sense and come into one experience that we're having. So that's parietal lobe. We've got 2 more. The occipital lobe is really important in vision. So this is at the back of the brain. And all throughout here, we have visual areas starting down here and then moving out laterally, moving out to the side, and then in towards the front of the brain, moving rostrally towards the front again. So that's the occipital cortex is primarily vision. And then finally, we have the temporal lobe, which sits just above, or right about where the ear is. So your ear is like right there. Let's see if I can move that a little bit. And that is important in, I can just click on it, I think. Oh, where did it go? And that is important in processing audio, processing sound, as well as memory formation. That's kind of the biggest thing that this does. You have the hippocampus, which we talked about right in here, right in this area. Yeah, you can see it somewhere around there. And then a lot of the memories are stored in here. You have semantic memory, and so semantic memory is like how to do things. And then you have episodic memory as well, which is more in the front, which is remembering events. All right, name the lobes. What is this one? What is this lobe here? Anybody? Yeah. Temporal? Excellent. Candium. How about this one?
Parietal.
Very good.
You take it first.
Parietal. How about in blue at the back?
Sorry, I have to make you work a little bit for it. Yeah, I have to move 1 step.
How about in blue? Anybody?
Occipital.
Yeah, you're allowed to. That's good. Occipital, perfect.
I wasn't because it'd be slightly unfair.
And finally, in the front? Very good.
I'll just take the candy that fell out of my reward.
All right. So now we're going to go in another layer to talk about more specific areas and talk about language and speech, because that is so important to humans, how we think, and obviously in social behaviors as well. So there are a couple areas that we want to pay attention to. And by the way, I know that neuroanatomy is a little bit dry. It'll get better when we get to neurophysiology. We just got to get through some of this stuff here. So, but for this part, for speech and language areas, we really start at the back here with the angular gyrus and Wernicke's area. So the angular gyrus is helping us, it connects to the visual areas, which we remember at the back of the brain, and it helps us convert that to spoken language. Because in the brain, we don't think in terms of written language, we think in terms of spoken language. And so this helps us sort of create that little voice in our head that is going along with us as we read or as we're going throughout the day and we're talking to ourselves. And that's closely correlated with Wernicke's area, which I've combined this other area in there because it's commonly just all known as Wernicke's area, which is used in language comprehension. And so this is putting together your consonants and your vowel sounds, and it's actually assigning meaning. It's talking down to your temporal lobe to figure out what those words mean and what our understanding of that is, and it's also triggering memories as a result of that. And this area here, this little pink area, is the first area where audio is processed. So it makes sense too that that's involved. And so this can kind of go up to Wernicke's area, or you can come from the visual areas and go to Wernicke's areas, depending on if you're reading or listening. Now, if you want to speak, that would then go to Broca's area, which is this large area in the front. And I mentioned that humans have a much larger Broca's area than other animals, and that makes sense because other animals don't really speak. There are some animals like whales and birds that do have an enlarged area here because they do have some vocalizations, but again, not as large as ours. This is typically involved in other animals in planning. And that kind of makes sense because if we're thinking about language, we can use language as a tool to help us plan. If we were just thinking in our mind, okay, I want to do this, this, and this in terms of pictures and not actually having a script to go off of, we wouldn't be able to get very far. But if we can use language and remember 5, 10 things to do, we have much more success with that. And so this area then later on connects to some of your motor areas up here so that you can actually make your lips speak and your vocal cords and all of that. And as we'll get into later, even though we're looking at little bits of localization, these are the primary areas where this processing is going on, but the brain is active at all periods of time and everything is kind of intermingled. And so you have sensory input that's coming in, obviously, and influencing what's going on here, influencing what your thoughts are, and then what you end up saying. All right, so the motor system, through your muscles is the way that the brain communicates to the outside world. This is sort of the only out that it has. And here are the most important areas that are involved in that. So we start with association cortex. And so what this is doing is it's aggregating a lot of our sensory information and trying to understand, okay, what do we want to do about that? I see a baseball coming towards me. I want to put up my hand and catch it. How am I going to do that? And so in terms of association, a lot of times we start back here with the PPC, the posterior parietal cortex. And this, again, I mentioned it before, but this is really important in integrating a lot of different senses. And then this decides in combination with the basal ganglia, which we can't see because it's behind the temporal lobe there. But these two areas decide, okay, I do actually want to move. And so that signal gets sent up here to the dorsolateral prefrontal cortex, the DLPC, which is up here in the front. And this part of the brain is saying, okay, what do we actually want to do? Let's come up with some instructions to tell the body, okay, I want to actually move in this direction. What is that going to look like? That goes to the secondary motor cortex, which is planning what limbs we want to use for that or what parts of the body. This is also really interesting for motor imagery because this is involved in planning. I've talked about motor imagery before, so this is imagined movement. That is all coming from your primary motor cortex. And if you've heard of mirror neurons before, these are neurons that are firing when we're watching someone else do a motor action. We're trying to do that ourselves. That is all in the primary or the secondary motor cortex. One little fact that I think is sweet that I like about this is when you are talking to someone else, you are subconsciously trying to mirror their facial expressions so that you can understand what their emotional state is. This is very important for humans to do. And even if you can't see your own face moving, if the other person can't see your facial muscles moving, there are still, your neurons are still firing and causing the muscles to move just a little bit. And so it's been documented that couples who have been with each other for a long time, say like 40 years, people start to say like, oh, they're starting to look similar, or they look more like each other. And the reason behind this is because they've been mirroring each other so long that the wrinkles in their face actually start to resemble each other, which is something that I think is kind of sweet. And then finally, we go to the primary motor cortex, which is the last stop in the brain. And this is correlated to very specific muscle groups, and this just executes on the action. So we've planned up until this point. We decided back here that we wanted to move. We planned on what we're going to do, sort of set those instructions, and then now we're executing on it. Once these fire, there will be a motor movement. And so again, we can just see that going through here. So we get to the primary motor cortex, we go down through the brainstem, and then we are kind of out through the spinal cord communicating with muscles. But the really important thing here is we see a lot of these arrows going in two directions and coming back because everything is in constant feedback and constant talk back with itself to make sure that I wanted to move my arm out here, I actually did move my arm there. And of course the cerebellum is involved in there as well, even though it's not depicted in this diagram. Last thing on the motor system. So we have this model called homunculus, which we'll see much better in the sensory system. But this is showing primary motor cortex. Again, you have the area where the neurons are, and then these are their connections, which we'll talk about later. But these are super well mapped because during surgeries, the brain actually does not have any pain receptors. So you can have someone who is awake in surgery and actually be prodding around in their brain and stimulating their brain, which isn't the most ethical thing, but it had been done in the early 20th century. And is also still something that's done when you're implementing a device for clinical purposes to make sure that it is actually working. And so from that, we've developed a really accurate map where we know, oh, if we stimulate this area, we'll make their knee kind of kick out, or if we stimulate here, their mouth will open and close. And so that's really interesting, especially for prosthetic purposes. If you want to make a prosthetic arm, well, you're going to be targeting the arm and the hand area. And if you can collect that signal appropriately, you could theoretically have a perfect control of a hand. And this I'm not going to spend a lot of time on because it's pretty confusing. But basically, in each of these areas, there are little populations of neurons that prefer a certain direction. And so instead of coordinating a specific muscle fiber to move, they just kind of decide, I want to move my arm or this finger forward. And when that's decided, the neurons that like that direction, that like 90 degrees, let's say that's forward, they fire, and the ones that are counter to that are very quiet. And so you can see there isn't firing on this side. And when I say firing, that just means activation. That's their go signal. So this is sort of how the motor system works in that way. It's little populations that decide what direction to go in, and then the spinal cord coordinates it from there. How's everybody doing so far? We're good? Yeah, go ahead.
General thing specific to this, but I've noticed that a lot of things are like not really that physically close to what they're controlling and taking input from, like the eyesight, like the brain at the back, and like the motor cortex is just not. Is it like just not a big deal to move information around that it's not a advantage to have things close?
Yeah, so it is an advantage to have things close in some sense because there is a limit to how fast a neuron can fire. It's not like electricity like we normally think about it, where you can theoretically have electrons traveling at the speed of light. We're working with ions, as we'll talk about, and the absolute max speed is 268 miles per hour that an action potential can fire. So you're right that neurons closer together would make more sense. A lot of it has to do with just evolutionary reasons in terms of how the brain develops. So the brain kind of comes out of the spinal cord and then it develops the brainstem and then sort of wraps itself around. And so areas that used to be closer are now farther apart. But then there are other pieces as well where with the visual system, it really is using almost the entire part of the brain, almost the entire forebrain, as a part of the visual system because of how visually focused we are as humans. And so in that sense, it's so interconnected that it actually does make sense because the thalamus, like I said, is a relay station. It's at the very center of the brain, and everything is constantly going through that. And so it actually makes sense to have it set up that way. But I'll get into that a little bit more later. Cool. Any other questions? Good? All right. So, we're still on motor system, very quick. So once the signals come out, they come down through the spinal cord and they exit between the vertebrae. And so they exit through this little area here, and then they innervate, they connect to muscle fibers. And so this is really important for anybody using EMG. So I know the prosthetics team is. The reason why we get such a big signal is when you're recording from an individual neuron, that's a really small signal because the neuron is tiny. But an individual neuron can innervate, connect to multiple muscle fibers. So it could connect to, I think it's like up to 50 or something like that. And so if we're connecting to these muscle fibers, which are massive in comparison to the size of the neuron, and it's controlling multiple of those, the electrical signal that we get is huge compared to what you would get from just collecting signals from the neuron. And so this is a summed response too. The neuron is pulsing, 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 pulsing. The more it does that, the more contraction you get, and the stronger that contraction. Cool. All right, somatosensory system. So this is your sense of touch. It's also giving you information about pain and temperature. Proprioceptive information, where am I in space? The reason why you can still touch your nose even if your eyes are closed and just sort of feel where you are in space. And then balance. And then we won't talk about the last one because that's just like internal organs and stuff like that. One thing that's kind of cool about your proprioceptive sense is that you have the actual information coming in from your body, right? And so we have information coming in from our muscles telling us where that is. But the brain it can sort of guess at this as well, which is why we get very good at using tools. So if you imagine yourself in a car, once you've gotten really used to driving your specific car, you kind of know where the ends of your car are, right? And so you just kind of intuitively know, oh, I'm in this space, or I'm like this close to hitting the wall, or whatever it is. And that's because your brain is actually able to incorporate these other things into its model, into its proprioceptive model, which is really, really interesting. So just quickly on this part, out in the periphery, we have these things called dermatomes. This is just where individual areas of neurons are collecting information from, and so these are all represented up in the brain for where they're collecting signals. So this travels up through the spinal cord, jumps across, and gets into the primary somatosensory cortex. And there are two different pathways, but it doesn't really matter. And so, yeah, proprioception in muscles. We have a feedback loop here. You have spindles that are actually in the muscles, which look like a little slinky. And as they get pulled out, they send electrical potentials back to the spinal cord. And the spinal cord pays attention to this, and if your muscle gets too extended, it causes a reflex to flex the muscle, and vice versa. If it's too flexed, it causes a reflex to relax it so that you don't damage your muscles. But this same signaling then goes up to the brain so that we can tell where we are in space. All right, so the vestibular system, which gives us our sense of balance, is also part of the somatosensory system. And so does anybody know where this is here? Yes, in the ears. Very good. So this part of the ear is the cochlea, and this is what helps us hear. But the rest of the ear is dedicated, or the rest of the inner ear structures are dedicated to having us keep a sense of balance. This is a really, really complicated and important system, and we don't really think about it until you lose it. If anybody knows anyone or has vertigo, it can be incredibly dysregulating to not have that sense of balance. And it affects other things too, where it can affect your eye movements and make it harder to see and have vision, and it just has this cascading effect because of how important the vestibular system is. So how this works very basically is, if we go back here, we have two main parts. So we have this utricle and saccule. Don't worry about the terms, but these little parts in here that detect forward and back motion. And then we have these semicircular canals, which are these big loops that try to cover your different directions, your forward, back, your yaw, and then rotational movements. So how that works is the utricles, the part that are going forward and backward, you actually have these little crystals in your ear called otoliths. And these sit up on top of this gel, and when you move forward, all of these little stones actually move forward in your ear, and that compresses this part of the gel, and then these sensory receptors down here let the neurons know that these otoliths have moved forward, and same thing when it goes backwards. Similar idea here. You have fluid in each of these semicircular canals, and then as you rotate your head back and forth, there's like a little flap that gets pushed back and forth, and that's how it determines that. And so it's really important to have functioning semicircular canals and saccules on both sides, because they counterbalance each other. When one goes forward, the other one goes back, and the timing between these is very important. All right, and so all of this information then ends up in one place. It ends up in the primary somatosensory cortex. And this over here on the right looks very similar to what we just saw in the motor cortex. This is the sensory homunculus. Same idea. Neuroscientists have been kind of poking and prodding at people's brains and noticing, oh, their thumb moves when we do this. You can do this non-invasively too, using a technology called TMS, where you can actually see parts of their body move when you stimulate specific areas, which is a little scary. But very, very useful in terms of when someone has an issue where maybe they lose feeling in one area, you can know where that is, and theoretically we could intervene. So I promised that we'd see a scarier image of homunculus. So the guy who discovered this, Wilder Penfield, had an epiphany moment when he discovered this, and he ran to, I think it was like the maid who worked in his house, because he knew she could draw, and he was like, you cannot leave until you draw this homunculus weird man. And she tried for hours and went back and forth, and this is ultimately what he was happy with. This is a 3D version of that. And so what this is showing us is that there's an overrepresentation of some areas in our body in terms of what we are feeling from. And this makes sense. So if we go back here, the space dedicated to things like the lips and the face and the hands is going to be a lot larger in respect to the actual size on our body, because we have a lot of specificity in what we can feel in our hands and our lips than we do on your back. And so these are called receptive fields, and they're much smaller, and there are much more of them on your hands. And so again, this is just a really clever visualization to show what parts of the body we prioritize getting sensory information from. All right, vision, the complicated one. This one, I'm trying to condense all of these down because they get wildly complicated, and I could do several talks on each of these, but so we've just got a few slides. But so light comes in through your eye and lands on your retina, which is the part of your eye that has all of your sensory receptors. And so your sensory receptors, as you've all probably heard, are called rods and cones. The cones are looking at grayscale light, and the rods are looking at grayscale light, and the cones are looking at color light. But they also both do edge detection and contrast as well, which is really important. This information gets collected here, goes down a chain of neurons, and then out through the optic nerve. And then it transverses through the thalamus, as we talked about. And here's another thing where it really is traveling the entire length of the brain and ends up in V1 here. And interestingly, we see that there is what we call decussation when it splits and goes on the contralateral, the opposite side. And so the entire left visual field that you have is processed in the right side of the brain, and the entire right side is processed in the left. And so it's not just like the right eye goes to the left or the left eye goes to the right, but it's really your visual field that is processed on the contralateral side of the brain. And the reasons for that are the way that the brain processes. So it's looking for, in the very beginning, very, very simple features such as edges and direction. And in each of these areas, it's looking for that separately, and then as it goes up the chain, it tries to combine them and make sure that they make sense. And so it's actually useful for it to process this separately so that it can check to see how correct its assumptions were when it gets up to higher areas. All right, so very, very simple overview of the visual system. It kind of splits into two pathways, the ways that we process this information. We have the where pathway, which goes up towards the top of the brain, the dorsal part of the brain. And then we have a pathway that goes to the bottom, the ventral part of the brain, which is looking at what. And so what is looking at shape and color. And then the where pathway is looking at where something is going and the speed of that. And of course, these are all hyper interconnected and they go back and forth. And this helps us put together features to determine what an object is and what it is doing, and importantly, helping us predict and base our actions off of the information that we're getting from this. And so these are all hyper interconnected, but the lower you go, starting back here and going up, the easier it is to determine what stimulus the brain is actually looking at. All right, hearing. So again, we see the inner ear, but we're going to ignore the semicircular canals now and look at the cochlea. So for sound, as you all probably know, we have compressed airwaves that are coming in and they hit here on the eardrum. And the eardrum vibrates these little bones here, which then press into the cochlea. And then when we actually get into the cochlea, over on the side we see these little bones again. And this is pressing against the basilar membrane, which is this thing here. So this is a cochlea that has been stretched out, if we can imagine it. So normally it looks like a shell. It has been stretched out into a long line. And this is placed very nicely. It's organized tonotopically, which just means in order. So we have very high pitch here and low pitch here. And specific sounds will resonate at different points. So if the sound is at this pitch, then it will activate, there will be sensory receptors underneath that will get compressed. And then the louder the sound is, the more compression there will be, the more force there will be that hits those neurons. And the softer it is, the less there will be. And this is why you damage your hearing if you listen to music that's too loud, because this basilar membrane comes down, it slams on top of your sensory receptors and actually breaks the tip off of them. And that's when you hear ear hairs and stuff. It's the sensory receptors that have little protrusions that come up from the top that get pressed down, which activates that receptor. And so this just kind of goes through the brainstem. Interestingly enough, again, to your point, it doesn't really make a lot of sense when the ear is right here. It ends up here, but it goes down through here, up through the thalamus, and then out. But this is the primary area where your auditory processing is. And that makes sense, though, because all of your language areas are localized there. Olfaction and gustation. So olfaction is sense of smell, gustation is sense of taste. These are a little bit more boring, in my opinion, but so we'll just spend one slide on them. There are chemoreceptors on your tongue. We'll talk about different receptor types in a little bit, but they can detect when specific chemicals are hitting those receptors, and then that's going to go up to the primary area that processes gustation, and then your olfactory bulb does your sense of smell. Okay. So how do we know all of this stuff? Well, one way you can do this is actually looking at the anatomy and the physical structures of the brain. And so in the early 20th century, there was a German anatomist. His name was Corbinian Brodmann, and he got under a microscope. He started staining brain tissue, and he made this little map over here and basically just drew a designation every time he saw a structurally different area. So there are thousands of different types of neurons, and basically whenever he saw a clear difference in the makeup, he would draw these little these little distinct areas here. And he was actually, and so here's just a more modern view of that so you can see it a little bit better. And a lot of this was pretty accurate in terms of functional specificity that we found as we've been able to do more science on this stuff. And so that's really interesting. Annoyingly, neuroscientists have all sorts of different names for every one of these areas and different ways of chopping it up, but this is still used. Example of that is the primary visual cortex. It's also called the striate cortex, and area 17 here. There's no reason you need to know that. I'm just annoyed about it, so I just wanted to get that out. But yeah, so that's that. Another way that we learn about the function of the brain is through lesions. So I talked about HM earlier. His hippocampus was lesioned there. So lesion just means removed in surgery. They did that because he was having epilepsy, which is progressive firing of neurons, overactivity of neurons. So they removed the original area to stop the epilepsy. His epilepsy stopped, but he was not able to make new memories, although he was able to recall old ones. So that was a really interesting case that taught us what the hippocampus did. And then, of course, now we have modern technologies such as imaging. And so over here on the left, we have something called a PET scan, which stands for positron emission tomography. And what this is doing is you're actually injecting radioactive tracers into your bloodstream, and then you're having your brain do a specific task. And that sounds scary, but it's not too radioactive, so it's okay if you do it once in a while. But basically, the parts of your brain that are more active absorb more of this tracer because they require more blood flow, they require more energy. And then this is really easy to see very specific areas that light up with this radioactive isotope. MRI gives us, which everyone's probably heard of, magnetic resonance imaging, gives us a fantastic structural view of what is going on in the brain, and it does that by, it has a really, really strong magnetic field that influences the polarity of hydrogen ions. Doesn't really matter, but that gives us information on the structure. And then functional MRI is also measuring blood flow, kind of similar to PET, but this gives us a more real-time understanding of what areas are active, and so that's just overlaid onto MRI. So that's very, very helpful and one of the biggest advancements that we've had in the last 30 years to help understand things about the field. The last way, which is more familiar to us, is using technologies that record from neurons. So this is something like EEG, which is on the outside, ECOG, which is a flat electrode strip that sits right on top of the brain, and then actual implants as well. And so what this gives us the ability to do is look at summed information of electrical activity, which is what we see here. Or if you're able to get a single electrode and actually stick it into a neuron, which we can do, neuroscientists do this all the time, you get these very, very nice spike trains that show, each one of these spikes show very clearly when one neuron has activated, which is really, really useful if we want to know specific function of an area. Let's say you stick an electrode into the visual cortex and you present some sort of a stimulus, you see the spiking correspond with the stimulus, so you know that it is a visual area. All right, I'm going to skip these because I don't think that how are we on time right now?
Feels like 22 minutes on with the room. Yeah.
Oh, okay. All right, yeah, we're going to skip those. All right, on to the more fun stuff, I think. So the neuron. The neuron is the functional unit of the brain that makes all of our cognition possible and pretty much everything else that goes on in our body. So there are three main parts of the neuron that I want to look at. This will be the last little bit of anatomy that we do. So over on the left, we have the dendrites, which are these branching structures that extend out, and these are kind of like satellite dishes looking to collect information from other neurons and collect signals from other neurons. And so these feed into the soma, which is the cell body. And that is actually, let's see. Yeah, I do have specific slides on that. All right, so to dendrites, the only other thing that I want to mention is that each one of these little strands, and there can be thousands of them, has these things called spines on them, and these are the receptors. And so if you really zoomed in, this is what it would look like. And this little line here is another neuron coming in and saying hello to it. So the soma is the cell body. If you've taken bio before, this should all look very familiar. This has all of the main components that a normal cell has, except it also produces special chemicals called neurotransmitters that can be used to talk to other neurons. And finally, the axon, which is the really exciting part. This is sort of like a wire that is transmitting a signal to another neuron. And the process for which it does that is really interesting, and we'll get into that in a second. And so it has the axon hillock, which is what is connecting to the soma, to the cell body. This is where when the neuron decides that it wants to send a signal to another neuron, that starts here. It shoots down the axon proper, the main part of the axon, and then it gets transmitted out here, and this is where it would connect to another neuron. These here, the myelin sheath, is not actually part of the neuron. These are all separate cells, but they act like insulation on a wire. So anybody who's done hacker projects before or worked with wires, you know that you want to insulate that both for your own safety, but then for the conductivity and speed of the wire. So that is what the brain is doing naturally there as well. It also protects it. All right, name the parts of the neuron. We have 3 parts. For candy, anybody? Is anybody still awake? Yes?
Like the last part, that's the axon is open, right?
The, which here?
Yeah.
This is all the axon, so you'll still get candy, but this is the axon bouton. The hillock is right there. So it goes hillock, axon, or axon proper, and then it gets transmitted out through the bouton.
I got them.
No, you're good. You're good. If there's candy around, you can get it whenever. I don't want to take any of this home, so whatever we're left with, people, please take. How about this area right here? The cell body. What is that called? Yeah. Yes. Very good. And finally, the satellite dishes. Perfect. Dendrites. Awesome. All right. So just quickly while we're here, you've probably heard gray matter and white matter before. I showed it on this thing. Gray matter refers to cell bodies, so this is the soma and the dendrites as well. And the white matter is the axons. So the white matter actually gets quite long. How long do you guys think the longest neuron in our body is? The longest axon? Take a guess. Throw it out there.
A few kilometers?
No. That's a good guess for the circulatory system, though, because that's insane. Yeah. So the longest neuron that we have is 1 meter, and it extends from the bottom of our hip down to our feet. And so that's a really, really long cell. It's the largest cell in the body. And the largest one in the animal kingdom is the blue whale, which is a 32-meter long axon, which is insane. Okay, so was there anything else I wanted to mention about? No, I think we're good. Okay, so there are other cells in the brain that are important to know about as well. They're called glial cells, and they make up more than half of the cells. In fact, some estimates have it up at 80 to 90% of the cells in the brain, and they help provide structure to the neurons because the brain is very, very mushy. The neurons don't really hold on to each other, so these cells help do that. You have microglia, which are really fun. They have these appendages that go out, and you can actually watch little videos of them kind of crawl their way through the brain. And they look for pathogens or dead neurons, and they just engulf it, very similar to a white blood cell. So that keeps everything clean. The astrocytes are sort of your handyman in the neuron. They help repair when blood vessels break. They help build scar tissue. They also help neurons come together. And they transmit neurotransmitter, which actually causes neurons to fire as well if they want to. And then these last two are involved in the myelin sheath. So this is just making a signal transmit faster and more reliably down the axon. There are 2 ways to do that. The oligodendrocyte is the thing in the central nervous system, and they have many protrusions for one axon. And then these Schwann cells are out in the periphery. All right, neurophysiology. So a neuron needs to be able to communicate with another neuron. How does it do that? It does that through electrical potentials. And so how they do this is they want to change another one's electrical potential to create a chain reaction that transmits a signal across a large part of the brain. And so neurons have to store potential energy that can be released when another neuron says that it should fire. And as we'll get into in a little bit, the resting potential, if you were to stick an electrode in a neuron that is not firing, is negative 65 millivolts. And this is really important because when sort of the floodgates open, this rises very, very quickly, and it creates a lot of potential energy. I'll talk more about that in a little bit. So a little throwback to chemistry. hopefully you guys remember some of this, but ions are atoms that have lost or gained an electron. I think we're all probably pretty good with that, but there is a valence shell, which is the outer shell of an atom, and they want that to be either completely full or completely empty. And so the important ions to neurons are calcium, potassium, sodium, and chloride. And so potassium has one more proton, so it has a +1 positive charge. Calcium is comparatively really positively charged because it has lost 2 electrons, has 2 more protons than electrons. And then down here, +1 and -1 for chloride. 2 more things that we need to know before moving forward are chemical force and electrical force. So chemical force is the reason why if you put food dye in water, it spreads out and everything becomes blue or red or whatever. It is the natural tendency for things to want to move away from like objects, move away from itself. And so if objects didn't have a charge, it would distribute, or if these ions didn't have a charge, they would distribute out evenly and nothing exciting would happen. But then there's also electrical force, which is the other element of this, which are magnets. We all like magnets. Magnets are great. So like charges push away, opposite charges attract. So for all of this to work, we need, each one of these ions wants to create a balance. And so it's not just that you have an equal number of ions on this side of a cell membrane or whatever the divider is, an equal number on this side. They kind of coagulate in different areas based on both the chemical forces and the electrical forces. And so if we have, let's say it's potassium, right? And maybe potassium is, normally it would be like this, that far apart, but for whatever reason, because of this would be just chemical force, but because there is electrical force as well, maybe it coagulates in one specific area. And so the sum of this, of whatever this ends up being between both the electrical force and the chemical force, is what we call the equilibrium potential. And so this creates a potential across the membrane, which just means a difference of charges. And each one of these ions has a preferred potential where if that was the only ion there, this would be the voltage across the membrane. So for sodium, and this part's very important, it is +60. So if we remember, neurons like to be at -65, so sodium is not happy with -65, it wants to change that. Calcium is really, really high at 137 millivolts. Potassium is at almost -100 millivolts, so it wants to drive that down. And chloride is pretty happy with -64 millivolts because normally neurons are at -65. Oh, and also there's this thing called the Nernst equation, which gives you this. If you care about it, it'll be in the slides. So again, neurons like to be at -65 millivolts because it creates a lot of tension with these ions, because the ions are not happy. Each ion wants the potential to be their preferred potential. So this means that when it's unlocked, there's a lot of potential energy force that can be opened up. So how are they opened up? How does this charge change? It is through ion channels. So ion channels are little protein structures that open and close to allow ions to flow in and out. And there are a bunch of different types that there are in the neuron that the brain has. So you have ligand gated, which just means chemically gated. So an outside chemical, or an inside chemical in some cases, comes on, it binds to a specific spot in the ion channel, and that channel rushes open and lets in whatever the specific ion is. We also have voltage gated, which responds to different potentials, which is going to be really important in a little bit. So as the potential changes from, say, -65 to -55 millivolts, that might trigger this channel to rush open. They're also mechanically gated, which is how we do the sense of touch. So these are all along, like all in your outer skin. When you actually press down, it forces the top part of these ion channels open, which allows ions to come in. And then there are some that are just leakage channels that are just always open a little bit. Maybe they randomly open and close, letting ions in and out. And initially, it doesn't make sense why you would want to do that. But yeah, Drake is helping me demonstrate this a little bit here. But what is current? Can somebody give me a definition of current? Flow of electrons, OK. Can we get a more broad definition of current? Because that's correct, but yes.
Flow of charge.
Yeah, flow of charge. But even think of a river, right? Current is something moving in one direction continuously. And so you can have a river that is current, you can have electrons that have current. And in this case, it is, yes, a flow of charge, a flow of ions. And so if there is no flow of ions, there is no electrical potential, which is why there has to be some leaking through. And what that creates is this thing called the sodium-potassium pump, which is just kind of recovering from that leakage and taking sodium and tossing it out and pulling potassium in. And that requires a ton of energy. About a third of the energy that your brain uses is just spent taking sodium and tossing it out and putting potassium in. This is possibly a little bit more of a useful demonstration from Crash Course, but we can see the sodium going out and the potassium coming in, and then that's just showing that we're maintaining this, this potential of negative 65. All right. So the action potential. Action potential is a synonym for the word firing. This just means that the neuron has activated, and this is happening in the axon. So this is an all or nothing event. It is a chain reaction. Once it starts, it cannot be stopped, at least not naturally. And it has to do with opening and closing of ion channels. Draw, in case I forgot. So I'm going to pull this up for a little bit. Cool. Okay. So let's imagine that this here is our axon. Good, I already can't draw straight lines. So this is our axon here, and we want a signal that's going from this end to that end. And so here is our cell body, and here is our axon hillock. And we have a lot of these ion channels that kind of go all the way down. We'll say that those ones there are for sodium, and we'll say that these little ones in the middle are for potassium. So K and Na. All right. So in the axon, these are the only ones that matter, at least in this part of the axon. And so what happens is the soma decides in the axon hillock, OK, I want to fire. I want to talk to another neuron. And we'll talk about that process in a little bit. And so what that does is it raises the membrane potential to above negative 65. And so if we were to record the neuron right here, so let's say that's an electrode. If we were to record the neuron, what we would get is a signal that looks like this. And what this is doing is right here, this is negative 65. Negative 65 millivolts, that's what it is at rest. Right here, we're going to draw a line at negative 55. Here is 0, and here is 120. OK. So what happens is once the soma is able to raise the membrane potential to negative 55 millivolts, so we've gone towards 0 a little bit. We call that depolarizing because it was hyperpolarized down here. It's depolarizing towards 0. Then at negative 55 millivolts, that triggers those electrically gated ion channels that we talked about before. And so we start with the sodium ion channels, which are these orange ones. And again, at negative 55 millivolts, these rush open. So these now open. And which way does sodium want to go if it's currently negative, I can't draw. If it's currently negative 55 millivolts inside the cell, and sodium, which is positive, wants it to be positive 60 inside the cell, which way is sodium moving into the cell? What direction is that? Well, so in terms of, so yeah, it's definitely going to be going this way, but this is the chain reaction going that way. If we have sodium ions here and sodium ions here, where are the sodium ions trying to go? Are they trying to go in, or are they trying to go out? And so remember, inside the cell right now, it's negative 55 millivolts. Sodium over here, which is positive, wants it to be positive 60 millivolts. So pretty big difference on the inside. Yeah, go ahead in the back.
So it's trying to go in because it doesn't want to stay in the light area. So there's, the same stuff. So it wants to go in because there's less voltage.
Okay, what were you going to say?
I was going to say it would go in because the voltage is lower than that one.
Good job, guys. You're both correct. Perfect. So yes, so exactly. So it comes in and it rushes in with a lot of force because it wants it to be positive inside, and it is positive. It's a positive ion. So this is now rushing in, and that is shooting our potential up towards 0. And so this is flying open, and this starts to happen all the way across like a chain reaction, right? Because as it raises here, as this gets closer to 0 here, now this one's going to trigger, and that's going to cause sodium to come in, and then this one triggers, and so on, and it goes down the chain. Once it gets up here, now it's overshot because sodium wants to continue to go up to positive 60, but now the neuron is like, hey, this is too much. We got to shut this down. And so what ends up happening, I'll draw it over here, is if this is our ion channel, it has this little magnesium ball on it, which I think is interesting. And this gets lodged in here at positive 20 millivolts. So this completely inactivates these channels, meaning that it is impossible for sodium to continue to flow in. And so now now that we've gone up here, the neuron wants to return back down to its resting potential because now this chain reaction is propagating down because more of these sodium channels keep opening. So how do we get this down? Well, that involves potassium. So at 120 millivolts, at 20 millivolts, the potassium channel is open, and which way is potassium moving, in or out? Yeah.
In.
Why in?
Because it's a lower voltage than attractive iron.
It's going to be, so I see where you're going, but it's going to be the opposite of that because, so potassium is a positive ion. So if it goes somewhere, it makes it more positive, right? And it, for whatever reason, wants it to be negative 100 in here. So it's going to rush out, and that's going to cause a very, very rapid drop. It undershoots a little bit, and then as those sodium-potassium pumps start to pump, we start taking the sodium that has now been in here, we push that out, and we take the calcium that has gone out here, and we pull it back in, and that's what keeps us at negative 65 millivolts. All right. So this is how signals propagate through axons. This is how sort of electricity works in the neurons. But yes, do we have any questions? Go ahead.
So the negative ions want to make it positive, and the positive ions want to make it negative?
It's confusing. Yeah.
In the area.
Yeah, so they want to make it within the cell, right? So there's also what's outside of the cell as well. And so it's not really discerning between that. Where we're getting that number is the equilibrium potential, which is a sum of the chemical force and the electrical force. And this is just what it ends up being. So all of these, again, if there were no other forces, if there were no other ions, sodium would equalize there, calcium there, potassium there, and chloride there. Yes?
I want to let you know we're 1 minute off the inverter reservation.
Okay. Well, yeah, I mean, people can go at any point. I know I don't want to keep you guys here for a long time. There aren't too many slides left, but seriously, like, I won't be offended at all if people have to go. All right, so I'll just say that. I mean, are there other people coming in here? Is that why you say that, or?
Yeah, should be good.
I think we're good, right?
8 o'clock, I'm assuming no one's in the reserve at like 7:30.
Yeah. It won't take me too much longer, but again, I just want to say, I know it's Thursday night, people are tired, there are classes, this will be recorded if you're interested. But yeah, so that is that piece. Oh, good, that's not going to work. Are we good with action potentials? Does that make sense to people? Good? Okay. This does not work super well. But I don't know, I think that the neurophysiology stuff is very, very cool because it's sort of creating a natural wire in its own way to transmit electricity. And the next part that we'll get into is how this actually translates to talking to other neurons. This is just a little animation of what that looks like. So this is running down the whole cell. One thing to mention is these chain reactions are only happening in between the myelin. So the myelin, again, are these, yeah, please take candy on your way out for sure. Just like dump it in your bag, I don't care.
All right,
Nice.
What about first? I don't know if I get the right thing for you there. You want more? There's 20 here. No, that's good.
All right. So one thing to mention is that this chain reaction that I've been talking about with all of the ion channels, the ion channels are only in between the myelin. And the reason that you want to do that is that it's a slower signal every time that it has to cause that chain reaction again, because it pauses a little bit at each one of these. And so you use the myelin as a way to prevent ions from leaking out on the way down the axon. And so just like electrical shielding, this helps speed up that connection. But the signal peters out over time, it gets more diffuse, which is why you need to keep refreshing the signal at each of these nodes. Those nodes are called the nodes of Ranvier. All right, so once the signal comes down the axon, we have to now connect to another neuron. And so this would be the dendrite of another neuron that it's connecting to. This is the synaptic bouton, the axonal bouton. And signals are coming from here to here across an interneural space, so just empty space in the brain, that we call the synaptic cleft. So that's what this empty space is called. And so as the action potential, can I get this higher? Yeah, there we go. As the action potential is coming down, now it's time to trigger the release of chemicals called neurotransmitters, which again, people have probably heard of: serotonin, dopamine. There are tons of them, but those are the ones that people have probably heard of. And it has to trigger that to go across, because that is actually the messenger that causes the next neuron to fire. So we have these little packages of neurotransmitter. So we'll say that this is a neuron that emits dopamine, called a dopaminergic neuron. And so these all have little chemical compounds of dopamine in them, and there are a ton of them packed in there. And so when the action potential comes down the chain, what's now down here are ion channels that are electrically volted for calcium. And if you remember, calcium was at, I think it was 123, a positive 123 millivolts, so it really, really wants to get in there. So it wants to make this very positive, and it rushes in, and now we have a tremendous amount of calcium in here. What calcium triggers is these little hooks that come up, and they grab these vesicles, and they pull them down, and then this becomes a part of the cell wall and kind of releases the neurotransmitter into the synaptic cleft. And these, just through random motion, find their way down and bind to these receptors down here, which are ion channels. These are the ligand-gated chemical ion channels. And so when that happens, these all open up, and we have a rush of likely sodium into the space, and that causes another action potential to start forming down in the next neuron. So does that kind of make sense? We're good? But now you have all of this neurotransmitter kind of floating around in this intraneural space, which can be dangerous because it could just keep activating this neuron over and over again because these don't actually get absorbed. So what usually happens is there are a couple ways that you can get rid of that. You either have those astrocytes that we talked about before, which kind of just eat them up, or another way is to use this little channel up here called a reuptake channel that just sucks all of this stuff up in here and then repackages it, recycles it. And one interesting thing is if you're someone, or if you're treating for someone that has depression or anxiety, one common belief with that is that the person is not getting enough activation with serotonin. And so you target neurons. So there's a class of drug called selective serotonin reuptake inhibitors, SSRIs. This is like Prozac and fluoxetine. And what that does is it actually hugs this little reuptake channel and makes it so that these these neurotransmitters can't get back up through, and they stimulate this more. Anyway. So yeah, this is just an animation while I erase. We'll talk about learning and memory, and then we'll be all set. So we see these calcium ions coming in and triggering some sort of ion to come in down at the bottom, making the next one activate. Other quick thing to mention, there are some that do make physical connections called gap junctions, where it's just basically a continuous wire. We don't have a lot of these in the human brain because this does not allow for a lot of processing, which I will get to now. Since we're short on time, I'm just going to send this video out after, but this is a video from Harvard that kind of explains everything that I just did, and it's really, really great visually. Oops, there we go. Okay, so postsynaptic potentials. I wish that this whiteboard erased a little bit better. But now we've got, so here's our axon, and it is connecting to a dendrite here. So this dendrite may have many axons connected to it. And we're just going to go in time that we have gotten the signal across from this axon, and we want to see what happens. So here's the dendritic branch. This might be branching off in a bunch of other ways. Doesn't really matter. So when this dendrite receives a signal from here, just like we've talked about before, this is going to, in most cases, depolarize, meaning that we are moving closer to zero, creating potential energy in there. And so we have that same thing where you're starting to have a spike. But the dendrite does not have voltage-gated ion channels, meaning that when the voltage is increasing, you don't have that chain reaction that you have in the axon. And this is very intentional. This is how the brain kind of does processing. And the reason for that is for a signal to get down the dendrite into the soma and then eventually to the next axon, you need to have a lot of signal coming in here so that you can get a large sum of signals that is able to preserve all the way down the dendrite. So it's not enough for one axon, one other neuron, to communicate with it. You have to have lots to communicate. So you need to have these some signals where you have lots of axons coming in that are telling this neuron to fire. And on the other end, so the ones that cause it to fire are called EPSPs, excitatory postsynaptic potential. There are also IPSPs, inhibitory postsynaptic potentials, that do the exact opposite. And instead of making it go up, they want that signal to go down. And so these kind of fight back and forth with each other, and this is how a neuron decides if it wants to fire or not, and how it makes decisions based on inputs coming in. And so that's a difference with the gap junctions in that this is actually making a decision based on the signals coming in. And so that decision gets down here, and if it is able to raise this, like we said, to -55 millivolts, then it starts the chain reaction and communicates with the next one. So for learning and memory, We can still use the same drawing. Basically, the difference is just that we are creating more connections with neurons that are firing together and less connections with neurons that are not firing together. So if you have some friends and you ask them for advice and Bob gives you really, really terrible advice, you're going to be less likely to listen to him in the future, and you are going to give him less influence over what you have to do. But Sam gave you fantastic advice on whatever to do, you're studying for a test, whatever it is. And so you want to listen to Sam more in the future. And so let's say that this is the Bob axon coming in here. You're going to take away some of these ion channels because you don't want to listen to him as much. So that means that for him to get through, he has to really be yelling and sending lots and lots and lots and lots and lots of signal. But for Sam, what you might want to do is create more ion channels here so that when Sam says something, when Sam sends a message, the response here is larger because there are more ions coming in. You might also extend another spine up so that Sam can send another axon, and you can add tons of these. And the last thing you can do is tell the Sam neuron coming in to send more neurotransmitter. So all of these things contribute to a neuron's ability to listen to some neurons more than others, and that's kind of similar to weighting in neural networks. This is the process of doing that. So on a large scale, this allows neurons to decide what neural networks they want to be a part of, which ones are accurate, and then diminish the features that are not correct when things are failing. I think. Yeah. All right, we'll call it like right around there. So I'll just do a couple more slides. So all of these are linking up with each other, and you have some neurons that are inhibitory, which means that you want that. Which means that you have some neurons. There we go. Some neurons that don't want other neurons to fire, and some neurons that do want some to fire. So if we look here, this is a very, very simple circuit diagram that's in the thalamus, in a part of the thalamus called the LGN, the lateral geniculate nucleus. And so this is taking in input from the eye directly and synapsing on an LGN neuron. And that LGN neuron normally goes up to the primary visual processing area. This is that area at the back of the brain. But it also has these black inhibitory circuits, and from V1, a circuit coming back to inhibit it as well. And the reason that we do that is it is limiting its ability to continuously fire so that it can refine the signal coming in over time. Because you don't want one area to be firing too much, because that would create sort of like a white spot in your vision. And so basically it kind of acts like a clock where it says, okay, if I fire 5 times in a certain amount of time, then I cut myself off. And this same type of processing in terms of using clocks, the neurons know how long it takes for an action potential to go down a specific circuit, and so they can set an inhibitory thing. This is where your circadian rhythm comes from, and your ability to coordinate over time is these sort of on-off clock circuitry. This is just showing a reflex circuit. Again, when it's fired too much, then the neurons tell it to be quiet. I'm not going to go through this, but this is just to show the tremendous complexity. I said that I simplified the visual system earlier. Each one of these areas is collections of millions of neurons of its own that is doing processing for your visual system. And so you're starting down here. LGN is in the thalamus, moving up to the visual areas of the back. Your dorsal stream is over here, going up there, and then your ventral stream is here. And so these are all of the areas that are implicated in vision. And the really important piece is that these all connect back to each other, because you have a lot of influence based on what you want to see versus what you are actually seeing and what is actually there. And so that's really important. Last piece is how we image neurons, how we see them, how we know what they look like. We'll actually start with this one. So this is staining. I have a book here in the front, my favorite coffee table book that is all of the drawings from someone named Santiago Ramon Cajal. He was the first person to be able to stain neurons, and so this is one of the first neurons pictured. And basically, you use a special type of dye that gets absorbed into some neurons. This is how we do it today. We use viruses a lot of the times, which is really, really interesting. So this is using the rabies virus. So rabies is kind of interesting in how it works. Let's say you get bit on the arm. What happens is the virus gets into your neurons and it moves in the retrograde direction, means it moves up, so from the axon to the soma to the dendrites. And it then is able to go across the cleft and continue to infect more and more neurons. And so it spreads like a tree up until it reaches into your brain, progressively killing neurons. So it's scary, but what neuroscientists have figured out how to do is they modify the genes of that to, one, cause neurons to glow green when they activate. So this is just like a regular photograph of green glowing neurons. And to make it so that they can only go back in time once and they can't continue to infect. And so what you're able to do is you infect a neuron downstream from this one, and this neuron is something that infected neuron is connecting to. So this is a really good way to determine circuitry. You start in somewhere like, let's say, the thalamus, and then you look in the visual cortex and say, this must be connected to here because this is where I injected, and these glowing neurons are the result of that. So these are actual images of what it looks like. But yeah, I think we'll call it there. That's enough for now, yeah. Cool. All right, thank you all. I appreciate it. Hopefully that wasn't too hard to sit through. But yeah, there will be a recording too if people are interested. I'm always happy to talk about neuroscience if there are questions. And there's a lot of candy, so please take it. I don't want any of it. So I might take some Twizzlers, but other than that.
That was a very good lecture. I do have a quick question for you.
Yeah.
Where are the three bones that are inside the ossicles? You glossed over it. I want to see if you know it.
I did because I don't remember what they're called. What is? Hold on, I'm thinking. I can get it. I know that they're the oscillatory bones, but I don't know what each individual one is called.
Come on, it's like I'll even take the English translation of them.
What is it?
Malleus, incus, and stapes.
Huh?
Malleus, incus, and stapes.
What's the Latin?
What they mean is the little hammer is stapes.
Okay.
The malleus is I believe it's piston, and the incus is like the saddle.
Right.
No, you like the strip saddle.
Okay.
Yeah, I noticed the laser there.
5:30 Dawn, Monday.
5:30 Monday? Yeah. Send over an invite. Probably be good.
For what?
Keyboard meeting with
Sure. Yeah. Sounds good.
One other thing, what's the area of the brain that's opposite on the other hemisphere of the Broca's area?
Oh, I should have talked about that. Yeah, that's I don't know what it's called, but that area is